What I'm going to talk to you today about is the National Variety Trial System and the change in the way that the data is analysed, and a few suggestions on how I would go about using that information to the best effect. Now, at the risk of going over ground that you already know about, the NVT is a system of trials for a range of crops to provide independent variety assessment data. There are 600 trials scattered around Australia, blah, blah, blah. The current system of analysis is a major change from what's happened in the past. It is a two-stage analysis, as it has been in the past. It's the second stage in this analysis that has changed, and the biggest change is actually the way the data is being reported. Stage 1 is analysis of individual trial data, and stage 2 is the across-sites analysis, which for years we as in the biometricians and the breeders involved in this process have been going on about, don't use individual trials data to make your decisions. Use the regional means. Well, I'm going to explain the changes to that advice. Okay, so why are we doing this analysis? Firstly, estimating individual trial data is not enough. You need it to be combined in an across-sites analysis, but all trials are not equal. They're not equal in the amount of error, nor are they equal in the way that they are predicting varieties. So we are changing the way that we do this analysis. And then all environments, and this is biometrics speak for all variety by trial estimates are not the same. And so what is happening is we now have a production value, which is essentially a modified mean. And it is weighted by the error and by the genetic correlation between trials. So what we are saying essentially is, by all means look at the regional means, but you can get a lot better information by looking at the across-sites estimates for individual locations. Now that seems like we're telling you to do something that we were telling you not to do in the past. But in fact, those numbers are not estimates from the individual trial, they are estimates from a group of correlated trials. Now, why are we doing it? Because the best prediction of performance from next year's, the best performance of estimate of variety performance for next year is not what happened in the experiment at that location last year. It is from a group of trials which rank varieties similarly for that region. And those trials are not necessarily the trials that are closest to where you are. And that seems like a strange thing to say, but nevertheless it's true. This analysis is world-class, world-leading technology for data analysis and presentation of data. Make no mistake, it is really good stuff. If you want a detailed explanation of the statistics, if that's your particular bent, then Alison Kelly, who is the biometrician who does some of this analysis, was here earlier. Ask her. I'm not going to go there. What I am going to do is actually go through the formats of the data that we're presenting and some of what I've done in trying to interpret this data for you. You can get data in three different formats. The individual trial data that I would suggest to you that you do not use for predicting next year. Look at it and try and figure out what happened in your region for last year, but don't use it for choosing the best variety. You're also going to see regional mean data. That's what you've got in the past. That's a reasonable estimate, but it doesn't give you the magnitude of the genotype by variety, genotype by environment interaction, or the variety by variety interaction. The production value, or the BLUP that we're talking about, is a much better estimate. And that will come in a number of formats. One of them is a graph in the crop variety sowing guide, and if you've got a copy of the Queensland crop variety sowing guide, you'll see some examples of that. The other one is that you can download an Excel tool from the NVT website that will allow you to put your selection of varieties and allow you to choose the site that you think is more closely matches where you are, and that will give you a range of performances. I would urge you to actually use what they're calling a BLUP in that tool, so that all of the yields are zeroed. So it's deviation from site mean yield is what we call it, but everything is relative to zero being the average yield, and you'll see what the significance of that. All of these presentations of the data are the same data, just in a different format to make it easier to see. This is Blue Poles, bought by Gough Whitlam in 1974, approximately 1.5 million, currently insured for 35 million or thereabouts. This is my version. It cost somewhere between 1.5 and 35 million to produce, depending on how you do the sums. And I put to you that it is worth heaps. This graph has every variety for which we have a yield estimate for North Star in New South Wales, all centred on zero. And the format of this graph is consistent throughout my talk, so I'll explain a bit. They all have plus 0.75 tonne, minus 0.75 tonne, and yield estimates from 2010 to 2014. If you do that for the sites in New South Wales and Queensland, you get a lot of information. You have to actually reduce that to make some sense of it. To do that, go back to the regional sowing guides, do what you've always done, get the number down to 5 or 6. And you do that by looking at stem rust, yellow rust, leaf rust, crown rot, yellow spot, etc. Coleoptile length, there's some discussion about that later. Sowing time response. And regional mean yield. That's what I do. That's what the individual trial data looks like. I like to colour code it using conditional formatting in Excel. If you're familiar with Excel, and that allows you to have a scan about what happened last year. Don't use that for your decision making. New South Wales regional yield trials data, reported in the crop variety sowing guide. That's where you start. I put it all into a graph just because I'm a visual person. Rule the line across at 100%, and then look at the varieties here that are performing and look at them in more detail. If you happen to have a favourite Spitfire or whatever it is, compare it, but compare it with the varieties that look like being better than the one that you're currently growing. If you do that, this is the graph out of the crop variety sowing guide that should have been in your satchels. This happens to be for Mungindi, and you can see that the graph has zero mean yield. It's the BLUP, and it's 11, 12, 13, 14. The varieties listed are under here, and you can see reasonably quickly here that Suntop is a relatively stable, high-yielding variety at Mungindi. Spitfire, moderate yield, sitting around site mean yield, but quite stable. And Sunmate, also high-yielding. This is the sort of information that you can get comes from the analysis. If you go to the Excel tool from the website, you can recreate that graph for yourself, and this is what it looks like. But over here, you can actually change the varieties. And so if you're not interested in these varieties, you put in the ones that you are interested in, and there's your comparison for Mungindi. If you think that you're at Capella or Surat or wherever it is in central Queensland, you simply change the region and then select the site that you want or the location. And by doing that, you can build up a picture for all of the locations that you think are likely to be relevant for your area, for your growing conditions. And it will give you an estimate of how stable and how high the yield are in that region, but it's not based on individual trials, it's based on this group of correlated trials. The reason that I say use the site mean yield or the BLUP centred at 0 is because this gives you a much more stable platform to look at to be able to compare varieties. This is the same data graphed by the experimental mean plus these values. And you really can't, I can't make sense of that, whereas this one actually looks sensible. The other thing that I'd note is if you're at Mungindi, it looks like you're going to have negative yields next year, so I wouldn't bother planting a crop the rate this is going. The comparison between these yield estimates and the actual yields from Mungindi in 2014, they are correlated. But that's all they are correlated. So what I'm saying is don't use the individual, what I'm saying is that these yield estimates, while they look like they're reporting individual trial data, they are not. They are actually reporting the data from that group of correlated trials. Sometimes they're correlated more closely than others, and that is exactly why I was saying earlier that not all trials are equal and not all environments are the same, and this is a reflection of that. Now, I would like, how are we going for time? I would like to actually go through some observations from the analysis, and these are observations that I find interesting and that I hope you will too. This is each of these little boxes is basically the same graph that I put up earlier, the blue poles too, with all of the other varieties stripped out. So I'm just reporting Spitfire and Suntop. And it becomes pretty obvious fairly quickly that in all the locations in northern New South Wales that we have estimates for, Spitfire is higher yielding than Suntop. And so that would make you reasonably confident that in fact not only is Suntop higher yielding, but its yields are pretty stably higher yielding than Spitfire. So if you're considering growing the two of those, you'd look at the other traits like screenings and tolerance to crown rot or whatever it is that you use, and use that as a factor. But this actually gives you an estimate of the yield penalty that you would be suffering if you were deciding to grow Spitfire instead of Suntop. If you go to Queensland and you start looking at the sites in central Queensland, then the differences actually seem to be shrunken. So here we've got Capella, Jeringa, Roma, Biloela, Springshaw, although Roma's actually in southern. So you can see that by using this information you can probably better tailor your decisions to the region where you are, knowing that this has all been adjusted for the errors associated with those estimates. There's a few other interesting comparisons. Well, I think they're interesting. Gregory versus Mitch. It's clear from this that Gregory and Mitch respond to environments in a very similar way. Those curves are all parallel, but that Mitch is higher yielding. There are sites like Nyngan where they're relatively close together, or locations rather like Nyngan, and locations like Merriwa where it's much more consistently, where Mitch is much more consistently higher yielding. You go to southern Queensland and central Queensland, and again, central Queensland seems to be a little different in that those two varieties, Springshaw or Biloela and Capella, they are much closer together. So the differentiation between those two varieties is much lower. You use this information to actually make decisions about whether you want to grow Mitch or Gregory. And there are other issues here too, like you have to factor in the AH versus prime hard issue. Another comparison that I thought was worth making was Gregory, Mitch and Viking. The Mitch and Viking being newer varieties, how do they perform? Well, there it is. They're all performing sort of differently, but in a similar sort of a way, so that they're all reasonably comparable, except that Mitch seems to be high yielding. Same in central Queensland, except you wouldn't grow Viking at Capella because 3 years out of 5, it's going to be costing you quite a lot of yield. Same at Springshaw, 2 years out of 4, it's going to be costing you. So you would actually be looking at growing Gregory rather than Viking. Spitfire, Sunguard, they're both stable varieties, moderate yield, and this data supports that hypothesis as well. So you can use this information to actually look at the stability and the differences between locations much more effectively than you can if you're just looking at the individual site years in the past. I've had a lot of help from Alison Kelly and Clayton Faulknell, the two biometricians, Peter Matthews and Alan Bedgegood. And this work is funded by GRDC, the NVT trials, and they pay me to do it.